Very good evening, everyone. Your most welcome, warm welcome to the June session of MBBT. I will be your trainer, Dr. Simranjit Singh, for the upcoming days. Very good evening. I can see a lot of students there. Rutha, Saurabh, Pratiksha, Yashvi, Kalyani, Poonam, Jyoti, Anchalam, many students. That's good. Beautiful, beautiful. Um, I think the day I had my first session, the introductory session of like 30 minutes, there were few students were there, and later many more students have joined me on the Telegram group. So the first session was done, let's say it was on 6th of June. Then there are many students have joined later also. So we will have that session a little bit more again so that things get gradually fine. So we were having, first of all, please confirm if my voice is fine. Is it clear and crystal without any disturbance? Only then we will proceed. And second, uh, all is good with the image. You can see my screen, because these two things I will ask every time before I initiate my lectures. Because if these two things are not working, it will not make sense to go ahead. Beautiful, beautiful. So first day started strongly, nicely. Our sessions will be also strongly and nicely in upcoming days too. Superb, superb. Yes. very nice students. Mhm. Yes, Diptesh Chakrabarti, that will be done in upcoming days. So at the moment, if I increase to 720p, the data will be a lot, will be consumed from my side. But I today keep it to this 480 so that we can have the smooth class. It's just this class is being there for the introductory class where we will be not going through much of the scientific work today. But gradually, when the time goes, we will make it happen to 720p. No worries. I know it's 480p now at the moment, but there is nothing that will interrupt your. No, no, no worries. That's good. So there was one conversation before we go ahead, that which platform will be the better for which platform will be better for these online classes. So I wanted to put this message, but I thought I will let the all class be there. Then I will initiate that talk. So as I have shared my link around 7:55 p.m. So it's been like not 10 minutes. So in this 10 minutes, around 88 or let more students be joining us soon. So what happened is in the Zoom thing. So I have all the platforms in my this small system. Um, so I have Zoom also, I have Google Meet also, I have Microsoft Teams also. We can have all the platforms I have for the online classes. So these sessions that have been going on, it's been like more than 1 year, these online sessions with Edufabrica. So according to my own experience with the students and to run it smoothly, so with the Zoom, it's maximum participants that we can go ahead with 100. But okay, we have till now 82. But if it goes more than 100, then we cannot have classes. Then students will be not added there anymore. And each sessions that we go in the Zoom, it's for 40 minutes. If I go for 40 minutes, then after the 40 minutes over, then I have to reshare the link again, and then all the students will join it again. That will take around 10 minutes time, 10 minutes window after the 40 minutes. Yeah, so that's 10 minutes is just being coming up, coming out, and the continue flow of the lectures, the continue flow of the presentations, it's all got disturbed. So in that sense, Zoom, for the long run, if it is 1.5 hour class or 1 hour class, the Zoom, who has the full package, that's the one of the best thing in the world. But at the moment, I don't have the full. It's quite costly actually. So maybe in future, things develop, I could buy that package, and then we can have longer videos. And second is Google Meet. In the Google Meet, yes, duration, there's no limit on the duration, but the problem is with the recording there. So I have a Mac at the moment. So in the Mac, I have downloaded some softwares, but they were not good enough to record for the like more than 30 minutes or 20 minutes recording sessions. Yeah, Harmanpreet Kaur. I'm coming on that on Google Meet. So sessions, there is also limit there for 100 students, not more than 100 students could join there. So that's one thing. Next thing is this third thing is this YouTube streaming. So in this YouTube streaming, what I found the best part is we can have as many as participants as we want. Yeah. Second, say it's not 92, it's getting more and more students are coming up. Uh, and second is the streaming, the streaming data that it takes. Yeah, the streaming data that it takes, it doesn't take as much as the other streaming that we are doing. So I have been living in the village for like almost 8 months in Kanpur. So everyone suggested me whenever the students were coming, because some of the students were coming from the remote areas, though doesn't have Wi-Fi at their house, and they are just with their 1.5 GB data per day, they're surviving on that. So in that sense, this YouTube recorded lectures for them, they were pretty good. And moreover, whenever I'm trying to record these lectures, so these lectures, they made this start of around 100 MB or 120 MB. So by this, at the end of the things that will be over, there will be around maybe 20 or 30 video lectures or more than that. And having those lectures, and I'm sharing this over this group, so over this Telegram group. So lectures will be pretty much concised within this group. So you will have the recorded version in this, and they will, the students had problem. That's what I'm saying. The students used to tell me, so they, most of their mobile data is being just filled with our videos. So you can't save them for the long on the long run. So on that sense, I have created this 10th June MBBT this group. Yeah. So in this case, so I have also created one playlist under the same YouTube channel, the June batch. So whenever you had any want to see any lectures or want to go through them back and want to learn about it again, so they will be there for you. And second thing, about live sessions, like live talking with each others. So after 10 or 50 minutes when the lectures will be going on, so I will be coming back to this window, and pretty much, believe me, so I try to satisfy each questions whenever they are coming, whether it's intellectual, whether it's technical, whether it's any personal or anything. So I will be there for you, so don't worry about all these things. So according to our own experience, we have chosen the best platform which will satisfy majority of the students here. So yeah, MST, is it Microsoft Teams, right? So yeah, Isha Tripathi, I had this also, this one, Microsoft Teams. So the problem is that sharing the recording lectures again, they will be, it will be concised just within your phone or in your laptop. So in that sense, but this YouTube will be there forever, and most of the students could log in anytime, and you don't need to like go through back, because at the end of the time, because our lectures are more than maybe it could be 30 lectures or 35 lectures in total. So that's why I'm saying, so it will be nice, we can have all them here. So okay, let's start with the introductory class today. First of all, I give my little bit of my own introduction. Who am I? Where it is. So from my education point of view, yeah, my schooling was done in Delhi. I'm from New Delhi, and I had my schooling from SS Mota Senior Secondary School with the PC, with medical and mathematics, and then went for my bachelor's in biotechnology in Shahid Udham Singh College of Engineering and Technology in Punjab. And then after finishing that successfully, I went for my master's in Germany, this Justus Liebig University, where I had my master's in agrobiotechnology. But as I was living there in Europe for almost 1.5 years, and during that time, what I saw that with the GMO crops, transgenic crops, all Europeans were pretty much against them. So they were not up to want to eat these new fields, new crops used with the transgenic technology. So in that sense, I just thought not to work in that line for my master thesis. So I switched to human medicine, and I work in the cancer field, where I have to find out endothelial specific molecules which are involved in the regulation of tumor cell extravasations. And after finishing my master successfully, I switched to my PhD in the same year, in 2013, in the next year, in April. And then the topic was something related with the heart failure. So I worked in the basic science in the redox regulations of PP1 and ER stress regulations of connective tissue growth factor in cardiomyocytes. So I finished successfully in 2017, was done with my studies, everything was over. Then I decided to come back for my to come back to India in 2000. Yeah, it was in 17 itself I came, and then I started my job at the in the August in Mata Gujri College, as a assistant professor. So I was in the part of biotechnology department teaching various subjects, and then afterwards that was over. Then I went for Edufabrica, where I was part of as an online professor. So I have joined this in last year. So I was teaching various subjects related to molecular biology, biochemistry, proteomics. So I brought all these that the sessions that you're having, enjoying this molecular biology. So last year in July, I brought these sessions with Vatsal Tripathi, Shivani ma'am, and Anuradha ma'am, to bring it this online lectures of biological series. So, and then I'm currently was working at the Rama University. I joined in November as assistant professor. But unfortunately, due to this COVID time, we also have lost this thing. So in this June, just actually yesterday, we were done with our job, and now I'm just fully dedicated to Edufabrica at the moment, with our MBBT course. And yeah, that's much of me. And then yeah, some of my previous projects works, I was also work as HR. So I had some publications out of that, some poster presentations, went for some conferences, some academic. That's not too much. So so anyone wants any help in the future of like CV making or resume platform, which template should be good? So I have used quite like number of platforms in order to make templates. If you need any assistance in regarding to make your resume more visible to the public, you can ask me anytime. I will be very happy to help you in the same. Second is, if you need any counseling or career counseling for which course you should go. I haven't asked you anyone. I will start asking now about your introduction. My doors are always open for everyone. In any way I could describe the which field you should go or how to go abroad, which university you should approach, how should the filter the things. So feel free to ask me. Second thing is, yeah, before I start taking your introduction, why I have created this MBBT course, actually molecular biology and biochemistry technique course. The main reason to do was that whatever the experience that I had in my, yeah, in my PhD or during my teaching experience. So what I feel as a student when I was studying in bachelor's and master's, that I was not getting the practical experience or virtual experience. So I was learning about southern blotting, I was learning about electroforces, I was learning about DNA microarray, I was learning about um, 2D gel electrophorese. So I was always reading them in the books, and we just thought in our imagine, okay, the gel electrophoresis, the agarose gel electrophoresis is happening in this way. We have this gel, we put this over the nylon membrane, and the bands get transfer over that. So this is how it was done, and we just write the same thing over in the exam, right? And we used to get scores also for the same, so that's no problem because grades are also important. But when I started to do all these things in my masters or in my PhD with my own hands, so things were pretty much drastic different, because when having the practical experience of all these techniques rather than just having the subjective knowledge, there is a lot of difference. Some of you might be doing these have some experience in that techniques already, and you know that once you start come up with, maybe let's say you started to do RT-PCR, so in the first go it will not go, by the second go you will get better, by the third row you get even better. So by the end of the week you will be master of running that RT-PCR. So that's how things develop. So this course is dedicated to teach you all techniques with virtual videos, with virtual experience, one by one. So whenever we do one technique, there will be one video about that, how this is done practically in the lab from the industrial perspective, and so that you can have them with you. Second thing is, let me share you the objectives. I hope you all, the objectives have been shared with you. Edufabrica, MBBT. Yeah. So this used to be a 10-day sessions before. Earlier it used to be 10-day session, but now we have switched to 1 month course. So if we are starting like today, officially we are starting on 10th of June. Most likely we will continue till 10th of July. So this is because we will have all and the sessions will be done from Monday to Friday. Yeah, sessions will be done from Monday to Friday. Weekends will be off. In any case, if I miss your class or by any chance I have to go or some emergency is there, so I will compensate that class over the weekends, or we can have one 2 hours class on the same day. So your classes will not be hampered in any way if some days is not there. So I will be responsible to take class on upcoming days. So if we are starting on this day, so we will be like having 1 week, 2 week, 3 week, and this 4 weeks. So around 4 weeks, we will have by the 10th of July, we will be done with this course. So about objectives. So maybe this is written day one, but this could go for 2 or 3 days also. So it's just to make some like necessary points that our MBBT course is divided into these subtopics. So on the first day, we will deal with all the basics principles of MBBT, your molecular biology and biochemistry techniques, like how the DNA is made, what is the basic structures of cells are, and then transcription and translations, your central dogma of life, all these things, RNA processing, all we will be dealing with them one by one, so that we can have a strong background of these uh, the basics. Based on the basis, we will slowly gradually go into the complex things. Then if we go into the lab, the first thing that you will see is the microscopes and cell culture. So your basically your whole molecular biology and biochemistry is based on these techniques. How good you are in the microscopy, how good you are in the cell culture techniques. So we will deal both of them in quite depth. What kind of media we should use while doing animal cell culture or plant cell culture, how should be the aseptic conditions we need to maintain in the lab, um, what are the criteria in order to have successful results at the end of your lectures. So all these things will be dealt in that. Then comes on third day, we will deal with some basics of your molecular biology technology. Then comes on the recombinant DNA technologies, like how we can make RDT, genetic engineering, cloning, and all these techniques. Then comes your day 5, where we will be dealing with protein quantification, separation, and purification. So all your chromatographic techniques, how with Bradford assay you quantify your protein and then you separate them with chromatography, HPLC, FPLC, gel electrophoresis, and then later you purify them. So all these will be done with proper like continuing manner. Then these electrophoresis, even though we know that um, the separations of how we make the separations between the molecules, but electrophoresis itself has been divided into many different parts. So far we might have heard about mainly agrosel electrophoresis, southern, northern, western blotting, but this might be very useful for you. Then the blotting technique. So this I always give a proper one day on this blotting techniques, because we always get confused that which one was southern, which was northern, which was western, southern western or northern western, zoo blotting, dot blotting. So there are many 6 to 7 topics are there. So we will dedicate on to that. Then this facts analysis are nowadays very hip, very useful technologies from diagnostic perspective, even from the other many other research perspective. So we will deal with that. Then bioinformatics, that is one of the key thing that we should also be aware that how this could be used in the molecular biology. Then on the last day we will deal with the applications of molecular biology. So whatever the things that I will be sharing with you, like just now I have shared um, these objectives. So I will be keep sharing them with you over the Telegram group on the same time at the same level, so that later they should not be, I should not forward or you should not forward, so that we're on the same platform. Okay, that was something about introduction part, how we will manage. And see, I have these, like, divided these lectures into these points. So I will continue them in the form of PPTs. So there will be PPTs, there will be videos, there will be some virtual labs. So we will continue with all of them, and all things will be shared. So all these presentations that I have. So once we are done with the lectures, we will be sharing with you over the Viber group or Telegram group. So that was the part of that, how the lectures will continue, how things will develop in upcoming days. The second thing is um, about the your assignments and review topics. So there are two things that I want to dedicate tomorrow actually for you, not today, because today is just want to give you a hint how the things will be going. So there is a thing called internship. So in internship, students have to write about a review article. So I will provide you the topics tomorrow about the same, that which topics you can write about the review articles. I will also dedicate one class on the how a review article is done, how you have to manage that. So we will do that tomorrow. So just don't be stressed at the moment, and you have to do this review article. So under during this 1 month period, but as the situation is like this, we might have to um, we might even increase the date for submission of your review article for 10 days or so. So based on your submission of your review article, only then you will get your internship letter. So we will share about that in upcoming days about that too. So no more discussion about review article, and this thing called assignment also, that you have to give an assignment that is not compulsory, but whatever we will be teaching you in the presentations, just write in a word pad about the internship. So these two things will be criteria also. And the end of the day, when we are done with the workshop, we will have an MCQ. Yeah, one MCQ will be there based on the all uh, these these uh, these workshops. So only the top 5 candidates with the highest grades will get the appreciation certificate. So we recently had that. Just a second. No, okay, I don't have. So there were 5 to 6 students who got that prize for scoring 30 marks. It was a 30 marks. There will be 30 MCQs at the end of the day, and you have to solve them in 40 minutes. It will be on the Google form, and the most score students usually get that appreciation certificate. So that's it, I think, from today. I should not put lot of burden on all of you. Otherwise, it will be too much on the first day itself. Okay, now, so thing is, once I'm done with my lecture, when things are over, I always come to your chat questions, and I answer them on the straightforward, so that we are on the same level. Sometimes, if I'm not able to answer any of the students' question, so you are most welcome to write me on the Telegram group personally, so I can answer them at that time also. So don't worry. Yeah. Okay, now let's take questions. So what was the internship part of it, as in that will continue beyond 10th of July? I want to discuss this thing actually. So I was told that there will be students who have joined for 1 month and students who have joined for 2 months. So I need to clear with my bosses how things are. So let me confirm with them how things will be. Yeah. So yes, sir. About the internship, will it go in deep in July? Deep in July? Varun Mahapatra. Yeah, it will be till July, or it could go further. So बच्चे Shalini, I will tell you about the review article Harmanpreet Kaur about the these things tomorrow. We will dedicate this date for the tomorrow only, so don't worry. Yes, as I have Kasi Aslan Ahmed. So as I said that these things will be shared on the Telegram, whatever I will be discussing. So don't worry about that. Okay, for MST Microsoft Teams, I can see how things are with it, so that we can have these classes there. So let me see, because you see, also there will be participant numbers, so we can see there's around 101 members are there. So that's the problem. So at the moment, we continue with YouTube streaming. Okay, so finally we had our class over YouTube. Yes. How can we reach out to you? Any career related query? I mean, Arunima Das. So earlier I used to be work from 9 to 4:30, but it's not anymore with me. So in the morning you can just ping me on my message on my Telegram. So I will be happy to solve you any query. Internship certificates, Shalini, will everyone will get it. So all the students who will, so what you have to do is in the internship, you have to write one review article. So there could be maximum 5 students could write it together, and you all have to submit them. And then the type top 5 performers is something else. It's about the MCQs. We have recently brought this part so that we can also have some examination part of it at the end of the course, and everyone should be joining us in that sense. So yeah. YouTube is better. Okay. MST is better. Thank you, sir. Okay. So I can see that some students at the moment is having problem with writing the questions in order to interact with me. Microsoft Teams allow we can do it on the Teams. Pratay Webhav. Okay. And what about the recording of the sessions? I'm just concerned about, because in my Apple I can't I don't have screen sharing like recorder in that. So that's the main thing. Okay. How about, so let's fix on the introduction. Okay, okay. So no worries. I will try to fix this. So some students are saying YouTube is fine. Some saying that this lectures even, so we will get these lectures even after the class on YouTube as recorded sections. Yeah, yeah, yes, that's the beauty of YouTube actually. So the streaming that we are having, it's almost like 35 minutes are over, and most of the lectures at the moment it will be there on there. And this thing, I don't have to, like, sometimes some students are muting, sometimes are unmuting, and I had a worse experience actually. But in this, if you're writing your question, just it's just getting out of your comfort zone. So everything has its ups and downs. Everything has its ups and downs. So let's do a voting here. So those who are going for YouTube, just write down YouTube is fine. And those who are going for MS Teams, just write down MS is fine. So we will make it done. We will be done with it just now. Just type in your YouTube, or are you in the with MST? Just type in here so that we can make a quick poll here. Okay, majority is YouTube is winning at the moment. Good, good, good. Hmm. So let's see. Okay, it's rushing quite badly. So what we can do that majorly that students want to have one-to-one interaction in order to understand the things. So I can do after one or two lectures, one session is over. I can create a 50-minute sessions after the 1 hour lecture is over, after 2 or 3 days, one MST session. Yeah. So this will solve the problem for everyone here. Saurabh saying by typing MST again and again, it will not make sense. You count one or more time. I understand you really, okay, Shivam, you want MST there. But as we can see that majority, everyone is happy with the YouTube streaming here. But don't you worry, we will continue. We will continue with the YouTube streaming. But after 2 or 3 days or 2 days, I will have a 50-minute sessions, a 50-minute sessions on MST. Yeah, 50-minute session on MST, and then we can have one-to-one talk about whatever the things will be there. Yeah, we can talk about that. Don't worry, we will also have that one. It's just these online lectures will be good for majority of the students. I'm saying this thing um, from your perspective, from the student perspective. I know from your benefit it will be good. Good. Then we are done. So give me quick your introduction. Give me your name. Shoot out your name, everyone, from where you are. Okay. So your name, your course, your year, in which in which semester or in which year you are, your place, and your hobbies. Go ahead. I want to know from where we are all belonging. Just and so that we can all be friends here. We can get to know about each other, and name, course, place, and hobbies. In which year you are. Just keep telling your complete information in one message, bache. In one message. Okay, Guinea Cooler. MSc Biotechnology first year, Delhi. Okay, most welcome. Ritika Sakre from Bangalore, fourth year. MBBS students, we are having MBBS. Okay. Tisha Jain, Bioanalytical Science, second year, Mumbai. Sneha Rajiv, currently studying Biomedical Sciences, first year from SIR HR Chennai, reading books. Good. Yashvi Rawat, live in Delhi, currently pursuing microbiology. Okay. Oncology. Oh, that's the most hip. Lot of research and lot of money is also there in that part. Indrani doing integrated BTech MTech from KIT, third year. Anurima Arunima Das from MIT Noida, MSc Biotechnology, first semester, Delhi. Prateek Mishra, Harmanpreet Kaur, MSc Human Genetics, semester, Punjab, reading books and music. Good. A lot of students I can see is from varied places of India and having BTech, MSc, even medical students are there. That's beautiful. Okay, it goes really fast. So I was at the Harmanpreet Kaur at the moment last time. Yeah, now Saurabh Singh, MSc Biotech, Pune, SPPU, artist reading books. Sakshi Sharma, Sakshi Biochemistry, final year, sixth semester. Aushi Shriya, Bhavesh Behere, Hind College. Okay, so Mumbai is there, Pune is there, Punjab is there, Delhi is there. A lot of students from Delhi I can see. Chennai is there. Wow. Gurugram, MSc Neuroscience. Mhm. MSc Biotech, Telangana. Welcome, most welcome, Rajeshekar Naidu. Okay, Sanjana Dubey, microbiology, drawing. Oh, microbiology students are also there a lot. Good. Nilakhish Patel, BSc Biotechnology. Shalini Mohapatra from Chandigarh University. Ah, CU, I was working there once, MSc Biotech. This is Prutha Parmar from Mumbai. Oh, B Pharma, tracking and painting. Isaac Lal, Kanpur, BTech, MIT Noida. Okay, I was in Kanpur also recently. I was going through this. I recently visited there. There's a JK Mandir, Parmat, Ganga Barrage. So I visited Kanpur a lot recently. It's a beautiful place. I really like it. Sandile Manlkar, B Pharma, Thane. Mhm. Diptesh Chakrabarti from Kolkata, first year MSc Biotechnology. Utkal University, Bhubaneswar. Most welcome. What about recording then? Shivam Krishwan, MSc Biotechnology, Uttar Pradesh. Most welcome. Shriya Praveshnan, BVSCH, a veterinary graduate. Uh-huh. Classical dancer as a hobby. Mhm. Gargi Day, MSc Botany, final semester, University of Kolkata. So Siani Jati, MSc Molecular Biology, second semester in Mysore, hobbies, cooking, reading. Vicky Kumar, BSc Chemistry, final year, Patna. Prishi Tiwari, Nagpur. Ashoka University, oh nice. Shivani Srivastav, Banasthali from Ghazipur, UP. Mhm. Anita Yadav, BSc Microbiology, Jai College. So microbiology I can see, some biotechnology I can see, biology I can see. I can see mostly so far from Delhi, Noida, Kolkata, Chennai, Hyderabad, Uttar Pradesh. Oh, it's unbelievable to see my whole India to be present in this class. Unbelievable. BSc Biotechnology from Shiv Nada University, third year. Mhm. Neuro-oncology and neuroimmunology, wow, they are quite complex subjects, and if you get a good grasp on them, Sarthak, Sanghinda, sorry, I can't spell that, Sanigida, sorry if I spell it wrong, maybe I can speak your name with the last name, Sarthak, that if you get the good grasp on these subjects, it's a very good field. Sonali Raj Tyagi from Delhi, Masters in Biotech from CU. I'm in writing, looking forward for PhD bioinformatics from abroad. Sure, it will be nice. There's a university in Denmark, not in Sweden. There are a lot of good universities in Sweden who are providing very nice courses for the PhDs in bioinformatics. Even in the Denmark, these small European countries, they're coming up very good. Isha Tripathi from Raipur, MSc Biotechnology, Vellore Institute, first year. Most welcome. National Institute of Pharmaceutical. Ah, okay. Naipur, Mohali, Punjab. I know this place. Sir, YouTube is fine. Yeah, there's a fight going on over YouTube and MST, but we will have a compensatory some mutual agreement. So everything is on the piece. Mahima Tiwari, Biotechnology, Banasthali University, Jai College, Microbiology, Shreya Anjusha Tirke, Shreya Tirke, BTech Biotechnology first year, Karuna Institute of Technologies, Coimbatore, Tamil Nadu. Momita Pradhan, MSc from KIT University. Nice. Shivam Kesharavan, Uttar Pradesh. MSc Biotechnology, first reading books. Pratishka Shrivastava from Kanpur completed MSc Biotechnology. Jayesh Survade, Naipur, oh, Aurangabad. Naika Bohra, course zoology semester 6th, Shivasagar, Assam. Oh, from our eastern part of India. Nice. Most welcome. Single currently BTech in biotechnology. Okay. MS team is fine. Okay. Yes, it is. Ayushi Shriya, BSc biotechnology, Ranchi, Jharkhand, playing badminton and writing. Hira Fatima, biotechnology, fourth semester, Gorakhpur. Oh, Lakshmi from MIT, BSc MSc dual degree, Kolkata, CSIR net qualified. Oh, that's good. Going to start my PhD. My best wishes to you, Smriti Gaba, MSc Biotechnology, Uttarakhand, writing. AITH, Kanpur, Biotechnology. Ritika Tandon completed MSc Biotechnology from Jamia Millia Islamia University. Oh, MSc in Molecular Biology. This is first time from Mysore, Karnataka. Applied Microbiology. Jyoti Chaudhary, BSc in Microbiology. Anjali Matthew, Biochemistry, listening to music. Misha Das, Biotechnology, dual degree, KIT University. Sanchita Naskar, okay. um, Sanskruti, MSc Biochemistry, first year. Pratik Mishra, BTech Biotech, second year, Varanasi. Oh, microbiology, Zoa, third year MBBS. Oh, there are 3 or 4 MBBS students also with us. Good. Because in my PhD, I have many. I was working in the hospital, so most of the doctors who were studying MBBS, so they were doing also research with us. So after your MBBS is over, you can choose to do only research, or you can do also both together. You can handle the patients and research together because that bring a quite nice balance for the future. So all the MBBS students who have joined us here, so keep in that mind, these techniques will be really helpful for your future career if you want to have both research and patient handling thing together in the hospitals. Madhvi Sivhare, a biotechnology third year from Banasthali Vidyapeeth. MSc Zoology, final year. Okay, so mostly I can see microbiology, biotechnology. Sorry, I cannot um, makes everyone's name, but I can see it's coming from all over the places. That's good. That's good. MIT, Noida, Rosie. Till here we have done. Okay, I have shoot out to everyone's name so far. I guess no one is left. Ramnika, second year ME Biotechnology, Bits Pilani. Okay, please carry doing with chemistry honors, Agastya Roy. I mean, so far I can see chemistry is highly used in the pharmaceutical background with the organic chemistry, by creating, synthesizing peptides, some new drug discovery. That has lot of future, because as a scientist, as a research scientist, even after like 3 or 4 years, you can find some really useful particles which were not identified before. This could be really make your career, the company that you are working with, could excite a lot of options. So just I will specifically would suggest to go for into pharmaceutical company if you are into the chemistry, or you can do go for some research institutes like ICERS, ISC, Bangalore, IITs. So there are a lot of research groups are working in order to find some really good and exciting questions. So you can try that one also. I'm Mahima Tiwari. I am from Uttarakhand. I'm pursuing BSc Biotechnology from Banasthali University, Jaipur. Oh, most welcome. BTech, MTech, dual degree, Amity University, Noida. Mhm. Would love to connect with you all. Yeah, that's the beauty of this group. So you are now open. So earlier we used to stop writing messages in that, but now we have opened it up. So you can. So I have shoot out all the names and all this chatting. So that's the thing. All these chattings that we have talked here, it will be saved on this YouTube platform. So whenever you will go, you can check which student was from which place, and you can talk to each other. So in the group also, if somebody is from, I can see many students from MIT. Maybe you have recommended each other to join this part. So you can, yeah, be friends here, make groups, and be friends forever. So Shalini Shukla, IIT, Indore, MSc Biotechnology, singing. Good, good. You're welcome. You're welcome. So, thank you, Sangam. This structured manner, it doesn't come in a single day. It takes some times. Anything that you want to learn, any skill, any habit you want to make, whether it's a waking 5 a.m. in the morning, that's also a structured manner habit. And this doesn't come in a single day. Yeah, this takes anything that you are learning. You're all belong to the life sciences, I can say. You're all students here that is present today in the moment in this introductory class. You all belong to the life science background. So you know much more about the human body, how the uh, systematically human body works. And this, that whatever you are learning, it doesn't come in a single day. So anything that you are learning or pursuing at the moment, it's come with a lot of perseverance, a lot of hard work, a lot of passion. And there should be also passion for that, believe me. If you are any of you students who haven't chosen this field of life sciences, and you're doing it just for the sake of parents said or somebody said there's no other course, you have to choose it, please try to think something else from now onwards itself, or think about how the next 5 years will be, because there will be some hard days where you have to like think, um no, I'm not good, i'm not I'm not being like born for this. But if you have a passion for something, you will wake up every day morning, and whatever that problems will be there, you will be good at troubleshooting it, getting success at it, and going up towards up towards the ladder of successes. So keep the passion, keep your morale high, and keep rising up. So never ever see behind of your life. Okay, so that was something about introductory part of the today. I hope I have shoot out everyone's name here, um, and everyone is doing good in their lifespan of your time. I always wanted to ask this question also, but this will take another 1 hour. That just think before you sleep or after this session is over. This is kind of your homework. That why did you choose biotechnology, or why did you choose microbiology? Why did you choose zoology? Why did you choose forensic science? Why did you choose any field, MBBS? What you're studying, and how this will be beneficial for you in upcoming 5 years. Just keep these questions in your mind, and that will solve many things for you in upcoming time. Yeah, so that's it for today from this part. Tomorrow we will meet again at 8 p.m. Yeah, um, and we will discuss about assignment part. We will discuss about assignment part. Just a second. We will discuss about assignment part and review articles, how they have to go. We will discuss with that. So what about IIT Roorkee, sir? You didn't mention anything. Dash can I do? What IIT Roorkee is better or not? Aryan Verma. I didn't get you, Aryan. Was this these certificates or this collaboration that you are in the part of this June batch? Was it collaborated with IIT Roorkee? Is it so? Because the Edufabrica, they had a partnership with IITs, yeah. So there are some IITs trainers also, but I have my own expertise. I am not from IIT, but I can teach you to my best level, to whatever the knowledge, little bit knowledge I had in my life. Of I'm at the moment 33 years old with around 7 to 8 years experience in the field of biotechnology. Um, yeah, so I will try to bring each part, each technique with the latest technology from CRISPR or anything. Yeah, so we will deal with that. So yeah, classes will be from 8 p.m. to 9 p.m. On daily basis. Yeah, so at the end of the, I don't know about IIT Kharagpur. Please consult with my higher authorities. Uh, that is it with the Roorkee. Maybe some of you might be with Roorkee, some of you might be with Kharagpur. But you will all get to connect, get a certificate with the name on it. Yeah, aims. Okay, so what I can see is there are 3 or 4 groups are being jolted together in this thing, in this group, and you will be at the end of the time is over, as per which course you have opted, you will get that certificate. So don't worry whether you have opted for aims or you have opted for them, IIT Roorkee or IIT Kharagpur, you will get as per that certificate. So no confusion about that. I can see there are 3 or 4 groups who have joined in this thing together. It's because of the collaboration with them. So all the faculty of the aims and faculties of the higher authorities, no, they are in touch with our uh, directors of Edufabrica. So that's how things are. Okay, any questions further? So I mean, at the end of the time, you will get one internship letter when you will write a review article here with us. That during this time you had a good experience. You have written on your own review article. You have been part of our journey during these. Learned all these basics parts of molecular biology techniques and biochemistry techniques. You will get a very full-fledged internship letter at the end of your session. So you can use this over the LinkedIn. I don't know if you're on the LinkedIn. LinkedIn is the best platform. Forget about Facebook, forget about Instagram. If you want to have a career, if you're career oriented, I would seriously say don't go on the Instagram is okay. I also use Instagram, but Facebook is going really bad. So you can be part of LinkedIn, and so some students have sent me request. So you can maybe in touch with me here also. So you can have really good career option, what things are happening in the field of biotechnology, how things are working, and this is professional, nothing any other stuff are coming up. So pharmaceutical companies, what is happening there, you can be in touch with the HRs of the same thing. So you can go into the my networks, want to check for, like, you go into the search bar. So let's say, let's clear this, clear history. You want to check maybe Glaxo Smith line, GSK. No, not like this. So you click on see results, click on people. So you will see all the people working in the GSK. Then you can type the location. You want to check in India, show results in India. So you can see a talent acquisition manager, the people who are hiring to work in the GSK, business executive, junior programmer, or something. You can also do, uh, yeah, many other. You can look for jobs, you can look for posts, you can look for companies. So all these things are available on this platform. So it's a very healthy thing to go for it. Yes, those who have paid for internship, they will get the internship letter only, not the others. Thank you, Saurabh Singh. Internship letter will be given to only those who have paid. Yes. So share IR LinkedIn profile. My ID. You can just type in Dr. Simranjit Singh. You will not doctor. I don't like to call as doctor sometimes, not most of the times actually. So just by this Simranjit Singh PhD, you can find me there. About publication, I will talk about that tomorrow. Don't worry, Sania Jati. We will talk about that tomorrow. No worries. Okay. Any further questions? Any queries, students? It will be Shivam Kesharwani. It will be in collaboration of both Edufabrica and IIT Roorkee. So I have seen like the some of the students, you can write them into your this 1 month training into your um, this what is thing called in experience that you are certified trainer in the MBBT, and you can put also upload that certificate here or internship letter. And just it's all about at the end of the day how much you speak, how much you are aware of these things, how much you can advertise yourself. It's all about showing who you are. So it's always good to be extrovert in these kind of things. I was very introvert in my BTech. I never used to talk with anyone. Just happy in my field, world of biological molecules, keep studying them and just stay there. But once you're into the field, once into the business, once you're into the game, you need to be very outspoken and friendly, socializing, speaking with everyone. You will learn all these things gradually. Some of these things are inbuilt by the character because of the family atmosphere or social friend atmosphere. Some genes things are also there, but gradually we learn on all these small things also. And students also, if you can just make like hit on the like, share, what is I don't these people are saying on this YouTube. Just subscribe to channel also if it is you can, because so far I have got a 948, so I need to make it 1000 or so. So it will be very helpful also. Okay, any further questions, students? And is it fine if our class go 5? You have any class after 9 p.m.? I hope you don't have any classes after 9 p.m. Our classes might go 1 or 2 hours also later, or not one, just 5 or 10 minutes. Okay, fine. So we can have our 5 to 10 minutes class if it is required of the time. We can do it also. Superb. On that note, I would say thank you very much. Wish you all nice evening. Yeah. So we are the one here in this session are the survivor of the second COVID um, layer, and I hope our near and dear ones were also fine during this time. No, no, we will have class from 8 to 9 mostly, but it could go just 5 or 10 minutes more, not more than that. So don't worry about that. So my point was that we are the survivors of the first COVID, second COVID also. We are the lucky ones, and let's make this to run further with whatever the God has blessed us with, and continue further in upcoming days. So we see us tomorrow at 8 p.m. Thank you. Goodbye. Good night. Take care.